Okay, so as an introduction to the alpha cell course, we gonna discuss the role of alpha cell in the pathogenesis of diabetes. So the discussion will center on the dysfunction of the alpha cells, which is the missing link or missing mechanism in the pathogenesis of diabetes that has been forgotten. Uh, but not lost. I will try to elaborate on this as we go on with the succeeding slides. So this is the cartoon pictures of the different islet cells and their arrangement. Um, as you all know, islets of Langerhans was discovered by four Langerhans in 1869, and the cytoarchitectures of the human islet were most of it is composed of the beta cells. It's about 71%, which shows association with other endocrine cells, suggests unique parakine reaction. The alpha cells, as you can see there, is our interest in this course. Beta and the delta cells, as represented in the cartoons. Other islet cells which are not shown are the PP cells or the pancreatic polypeptide, the gamma, sometimes called the gamma and the F cells. And the last one is the epsilon, the last that was discovered, which secrete gremlin. Now, let me just emphasize in that in the mouse islets, there's a little difference with human. The alpha cells are within the mantle in the outer border of the islets in the mouse islets. However, in human islets, the alpha and beta cells are mixed all throughout in the islets. So the history of glucagon begins with that of the discovery of insulin in 1921, as you all know, when Banting and Bess tested their first pancreatic extract in a pancreatectomized dog. Okay, this is the name of the dog, Marjorie. They observed that uh, the insulin-induced lowering of blood glucose was preceded by a brief hyperglycemia before glucose was lowered. And they thought at that time it was just due to epinephrine. Okay, they didn't bother to look for the reason and continue to discover successfully the first exogenous, which is the crude insulin, that have helped and saved the lives of tens and thousands of type 1 diabetes. And in 1923, Banting and Bess went on to receive the Nobel Prize for their work. Now, however, it was John Merlin, together with Charles Gimbel, were the one who discovered the impurity in the early preparation of insulin. They call it a toxic fraction causing a rise in blood glucose. So they coined the term glucagon from the two-word glucose agonist. However, it took a further 25 years before Sutherland and the Dube uh, established that glucagon is released from the alpha cells. And the identification of amino acid sequence of glucagon and the development of radioimmunoassay, a very sensitive glucagon test by Yalo and Berson, made it possible to quantify glucagon that paved the path for a more complete understanding of its role in physiology as well as in disease. Meanwhile, the contaminant stigma persisted among rank-and-file physicians, stigmatized for being largely diabetogenic in nature, with metabolic actions such as glycogenolytic, gluconeogenic, and ketogenic activities. And because of these reasons, glucagon was just relegated for the treatment of severe hypoglycemia shortly after sequencing and purification. The initial lyophilized glucagon that need to be reconstituted on your left uh, was quite inconvenient to use, especially in severe hypoglycemia. Uh, they developed an emergency kit wherein the reconstituted fluid is already in one kit. It makes it a little more convenient. However, in 2019, FDA had approved the first nasal glucagon powder for severe hypoglycemia that is delivered by as a dry powder spray in a portable single-use ready-to-use device. All of these are available in the States. Okay, glucagon, the pleiotropic hormones. Identification of a sequencing the 29 amino acids of glucagon, together with the discovery of a sensitive test, radioimmunoassay, have led to the understanding of these pleiotropic hormones or effect of glucagon, the effect beyond glucose homeostasis. Emerging evidence suggests that glucagon regulates a range of metabolic action, including lipolysis, ketogenesis, fatty acid oxidation, increase in satiety, and in decrease in food intake, uh, effect on thermogenesis and energy expenditure, and even bile acid metabolism. This glucagon-mediated improvement in lipid and energy metabolism may be quite desirable in patients with metabolic syndrome. Glucagon action and its signaling pathways was an area of previously underappreciated clinical importance. And with this new information, it paved the way to the renewed interest in the biology of glucagon and cellular aspect of glucagon secretion and action to those extending the concept of physiology and pathophysiology of the hormones from the experimental animals to human. Now, going back to insulin, the discovery of insulin was acclaimed as the greatest achievement in medical history and won a Nobel Prize within 1 year of its first injection into human. Since then, insulin has been considered the single most important metabolic regulator, and the catabolic derangement of type 1 diabetes have been directly attributed to insulin gap. The so-called insulocentric view of diabetes, and this has persisted for 99 years. Then in 1973, Roger Unger and Lelio Orsi of Geneva proposed the bihormonal abnormality hypothesis of diabetes, stating that glucagon elevation was as important as insulin deficiency, and glucagon was misjudged as an important pancreatic extract impurity, and glucagon was forgotten but was not gone. So in 1975, in his Bunting Memorial Lecture, Roger Unger presented evidence that elevated glucagon as the regulatory partners of insulin, the so-called bihormonal abnormality. However, the importance of glucagon in a normal glucose homeostasis and in a diabetic phenotype remain controversial. Clearly, the vast majority of clinicians and scientists continued to believe that insulin did it all and that glucagon had at most a relative minor role. Even today, they say even more few scientists or clinicians accept the glucagonocentric premise that the alpha cell dysfunction is the sine qua non of the diabetic phenotype and that its correction independent of insulin treatment would provide important therapeutic benefit, as suggested by Unger et al. In 2012. The sine qua non, which means without it, it isn't. Without glucagon, no hyperglycemia, even in the absolute lack of insulin. So here's the changing concept. or the changing dogma of diabetes. In 1922, the discovery of insulin, it was believed that lack of insulin is the key pathogenesis of type 1 DM, and giving exogenous insulin will solve all the problems of type 1 because lack of insulin leads to decrease in glucose utilization, increased lipolysis, increased proteolysis, increased hepatic glycogenolysis, increased ketogenesis, and decreased glycogen synthesis. In 1975, starting to recognize the role of glucagon since the different diabetes abnormality are caused by lack of insulin or by excess glucagon, the so-called bihormonal concept. In 2011, more emphasis on glucagon this time, and the premise is that lack of insulin directly causes some uh, diabetes abnormalities, uh, and insulin lack also leads to glucagon excess, which in turn causes other symptoms. Okay, so the so-called glucagonocentric. More emphasis now on glucagon. Now, these are the pictures of selected scientists with major contributions in the research of glucose metabolism, starting from Paul Langerhans, okay, and the last one was the last picture uh, is Roger Unger, who gave us the concept of glucagonocentric view, the importance of glucagon in diabetes. As you can notice, all but Roger Unger has years of birth and death. Uh, the last photo of Roger Unger was still very active, even very active in 2019. Uh, Roger Unger, who pioneered the concept of glucagon view, on endocrinologists who have contributed so much in the understanding of diabetes. He was born on March 7, 1924 in New York City, New York, USA, but passed away last August 22, 2020. Okay, so this was his work, the glucanocentric restructuring of diabetes. Okay. Here, they propose glucagon excess rather than insulin deficiency is the sine qua non of diabetes, and these are based on the following evidence. Number one, the glucagon increases hepatic glucose and ketone production, catabolic features present in insulin deficiency. Number two, hyperglucagonemia is present in every form of poorly controlled diabetes. Number three, glucagon suppressors like leptin and somatostatin suppress all the catabolic manifestations of diabetes during total insulin deficiency. Number four, total beta cell destruction in a knockout glucagon receptor knockout mice does not cause diabetes. And number five, perfusion of normal pancreas with anti-insulin serum causes marked hyperglycemia. So my task is to share with you the original work of Unger and colleagues based on these premises or evidences. Now, first, the study in animal model, wherein both mice completely depleted with beta cells without any detectable insulin and C-peptide in the circulation. Okay, the mouse on your right, okay, is obviously very sick with severe polyuria. As you can see, the ground was so wet with urine. Both of them carries or wear a pump. Okay, on the right, the pump contains just a buffered saline. On the left, the pump contains a glucagon suppressor to suppress glucagon effect. Okay. So the result on your right, the sick animal, because of lack of insulin, there's a marked hyperglucagonemia because of unsuppressed glucagon. Remember, this mouse pump contains only saline. While on the left, uh, who carries or the pump contains a glucagon suppressor showed a normal level of glucagon. So it appears that insulin lack is not essential for the development of diabetes symptoms. This difference was so remarkable that they explored this in further detail. Now, the study with a knockout mice or glucagon receptor knockout mice. On these mice, they measured both the glucose and the messenger RNA of glucagon receptor for the presence of glucagon receptor. So here, when they destroy all the beta cells by injecting streptozotocin injection, they use a double dose of SDC just to ensure all beta cells are destroyed. It shows no increase in glucose despite of no insulin from the destroyed beta cell, and so you can see total zero glucagon receptor messenger in the red box. Okay, and then you see the role of glucagon when they gave an adenovirus containing glucagon receptor injection back, and within 2 days there was a peak level of very high level and expression of glucagon receptor messenger RNA, but was only transient, which is usually the case. And you see eventually the expression drop off and disappear. And during the rise in the expression of glucagon receptor, hyperglycemia occurs, reaching to about 500 milligrams per deciliter before it went down to normal glycemia after the messenger RNA was no longer detectable. Now, this animal experiment do suggest that uh, what caused the syndrome of type 1DM metabolic consequences was not the lack of insulin directly, but instead the unopposed action of glucagon when insulin is absent. Now, this is the topographic scheme of normal human islets showing a unique or a spatial arrangements of alpha and beta cell, which is uh, extensive juxtaposition of beta cell in red and alpha cell in green that facilitates instantaneous insulin control of the glucagon secretion via the interstitial space separating the two cells, a very special paracrine effect. Now, to better understand this is to look at the interaction of islet cells in vivo in human. Normally, every beta cells are practically juxtaposed with alpha cells. And when you look at type 1 on your left, type 1 DM, obviously all beta cells are destroyed and the alpha cell sits in the islet themselves unopposed. The question is, what is the importance of this juxtaposition of the islet cells? That does it have functional implication or just by accident? And the answer is it has a vital implication in glucose homeostasis. This close proximity permits insulin to tightly regulate glucagon secretion and precisely control the insulin-glucagon ratio in healthy pancreatic islets. Now, here we look at the important paracrine effect in healthy pancreas in the islet isolate. When perfused with 5 millimolar of glucose, the beta cell secrete low level of insulin. This level does not suppress alpha cells. However, when perfused with 15 millimolar of glucose, that's 3 times the 5 millimolar, this amount of glucose, 3 times higher, suppresses glucagon secretion. When insulin is present, the alpha cell that is juxtaposed to beta cell receive the highest insulin concentration of any target cells in the body. The paracrine level of insulin reaching the alpha cells have been estimated to between 2,000 to 4,000 milliunit per ml. That's huge. So the insulin glucagon ratio with a 5 millimolar of glucose is computed at 0.4, and the high of 7.4 at 15 millimolar of glucose. Again, this is in normal, healthy pancreas. Now, let me try to explain this again through paracrinology, which is the study of normal insulin glucagon in response to glucose in the islet cells. In an isolated rat islet pancreas perfused with 5 millimolar of glucose, insulin and glucagon ratio is 0.4. At the bottom, you can see the red. When glucose was raised to 15 millimolar, insulin takes off in a dramatic spike. But look at the time length of that spike. It lasts only a minute or two. This may represent the first phase insulin response. The amount of insulin is less than 10% of the total insulin release, a small, very small compared to the total insulin release in the periphery. While it will have no effect in the peripheral metabolism, but inside the islet, it has a big insulin response because at this level, inside the islet, it suppresses glucagon release. So the very first insulin response had a profound effect on glucagon. The insulin-glucagon ratio of 7.4 is high, that tells the liver to suck up all the incoming glucose and store it as glycogen. And this is normal. Now, let's see now in diabetes. Okay, now this is an animal experiment in 1984, an isolation of normal rat pancreas and perfused with the constant level of high glucose of 25 millimolar. Okay? The measurement of glucagon coming out of the pancreatic afferent vein indicated some suppression of glucagon, as you can see that. Okay? Glucagon level as low as 1000 picogram per ml. Then a potent insulin neutralizing antiserum to neutralize the high 25 millimolar effect of glucose was introduced, hoping to disarm all insulin coming out from the juxtaposed beta cell. What they found, a 150% increase in glucagon level, and lasted only after an anti-insulin was withdrawn. The story tells us that the importance of juxtaposition of beta and alpha cell is to restrain glucagon secretion from the alpha cell. So insulin from the beta cell is the control of glucagon secretion. More recent studies show that not only insulin blocks glucagon secretion, but also the gene expression, as you can see in the slides. So, to test the key role of glucagon in type 1 DM, a study using glucagon receptor knockout mice, knocking out the receptor. The first graph showed normal mice after beta cell destruction in orange. As you can see, when you measure glucose, it's a typical type 1 DM. The orange curve looking at uh, normal mice after beta cell destruction, you see a hyperglycemic effect, okay, meaning type 1 DM, okay, showing severe hyperglycemia. The green graph represents glucagon knockout mice after beta cell destruction, meaning type 1 DM with glucagon effect, okay, so no hyperglycemia. Now, type 1 DM without glucagon effect, you see no hyperglycemia. Now, on the second graph, okay, is the measurement of insulin response to an oral glucose challenge. Now, they measured insulin this time. The knockout mice before and after beta cell destruction. As you can see, the orange knockout mice before beta cell destruction, you still have beta cell to secrete insulin. Okay, you don't have glucagon receptor, but you see insulin response after a carbohydrates challenge or oral glucose challenge. However, in a knockout mice after beta cell destruction and there's no insulin, you see very low or no insulin measurement yeah, measurement, you can see very low insulin level. However, now the the last graph is letter C showed the oral glucose curve of knockout mice. Knockout means no glucagon receptor before and after beta cell destruction. Okay. Remarkably, okay, although streptozotocin treated knockout mice, glucagon knockout mice, were incapable of secreting insulin in response to oral glucose tolerance tests. Interestingly, the glucose tolerance curve did not differ significantly from glucagon receptor knockout mice with an intact beta cell and a robust insulin response. In other words, insulin does not directly regulate glucose tolerance in a glucagon receptor knockout mice. Now, in fact, in every species and any form of diabetes, whether it's chemically induced, immune mediated, okay, by the use of alloxan or autoimmune, or um, in type 2 DM, in uh, uh, generalized lipodystrophy, and even in total pancreatectomy, um, there is hyperglucagonemia. However, many will question this. Many will question this glucagon-centric view in total pancreatectomy. The question is, where does the glucagon come from if you totally remove the entire pancreas? And in fact, this is the real challenge of the glucagonocentric view. It's difficult to reconcile because the question is, if you consider glucagon play a role in the pathogenesis of diabetes, so where's the glucagon coming from? Okay, so the experiment done by Unger and Orsi in mid-70s in a total pancreatized dog showing elevation of blood glucose, you see insulin level 0 and surprisingly very high level of glucagon even though the pancreas were no longer there. Where is the glucagon coming from? Measurement of gastrin vein glucagon was 3 to 4 times higher than in the inferior vena cava. So it seems that the glucagon is coming from the stomach. So here's they undertook careful search, doing electron microscopy and markers of ARPA cells. They found out homogeneous dense core of the secretory granules surrounded by halo of alpha cell found not only on the pancreatic alpha cells but also in the stomach cells. Now, when it comes to treatment of type 1 DM with insulin, the existing dogma is insulin therapy is sufficient therapy for type 1 DM. Now, we have insulin monotherapy for almost 100 years since the discovery of insulin. And we know our experience, we cannot duplicate the normal homeostasis of normal non-diabetic normal pattern of blood glucose on your left. Okay? Compared to a type 1 on your right, no amount of glucose load or exercise can move above and below the normal narrow range of blood glucose. Even in the best type 1 DM shows a pattern quite different from the normal pattern. Frequently low blood glucose causing severe hypo, while surge of blood glucose, particularly during mealtime, reaching as high as 200, 300. This is called glycemic volatility in type 1 DM. This is the pattern of life of type 1 DM, requiring multiple blood glucose determination each day in an endless chase for normal glycemia. Why insulin therapy cannot duplicate this non-DM pattern? Well, the difference between secreted insulin and injected insulin. Okay, the secreted insulin begins its journey in the islets of Langerhans. The juxtaposed secreting insulin called the juxtaposed secreting insulin cells to the alpha cells. Therefore, undiluted insulin suppresses alpha cells. Once insulin leaves the islet, it enters the portal vein. Diluted once it passes the liver, it gets to the peripheral tissues with further dilution. Compare this with injection to get the identical dose of insulin at the target level. And obviously, it cannot duplicate the endogenous effect of insulin secretion. So why not increase insulin so that enough insulin will reach inside the islet paracrine effect? However, you will only overwhelm the peripheral tissue and run the risk of severe hypoglycemia. Alternatively, one solution is if we can give a glucagon suppressor and reduce glucagon level without increasing its exogenous insulin dose and will not overwhelm the peripheral tissue and thus prevent hypoglycemia. The comparison of the itineraries and destination of secreted and injected insulin. This depicts the extreme differences between the pathways and targets of secreted and subcutaneous injected insulin. These differences may play a major in the extreme glycemic volatility that complicates insulin replacement therapy in type 1 DM. A, you can see secreted insulin, the response of endogenous insulin and glucagon to a high carbohydrates meal, the beta cell almost instantly secretes its first phase in bolus of insulin via paracrine route. Okay? The alpha cell respond to the insulin abundance by suppressing secretion of glucagon. The resulting insulin-glucagon ratio entering the portal vein is very high. This puts the liver in an anabolic mode in which incoming nutrients are taken up and are stored in macromolecular form. Insulin level in the posthepatic circulation are relatively low, with most of the insulin being bound to insulin receptors that are concentrated in the splanchnic targets. You can see B, subcutaneous injected insulin, the pathway and target of injected insulin are different. Subcutaneously injected insulin will remain at the injection site for about 10 to 50 minutes or longer. Thus, no suppression of glucagon is possible as the nutrients enter the circulation. The high glucagon level entering the portal vein will maintain fasting level of glucose production. When a large dose of insulin intended to correct the postprandial hyperglycemia finally distribute throughout the body, it will cause hypoglycemia. Thus, the roller coaster glycemic pattern may be the result of inability of injected insulin to reach its targets as rapidly as secreted insulin. Here are some of the published data looking at the level of insulin at different tissues. Highest at the islet and diminishing as it reaches the port, vein, and the peripheral vein, while the injected insulin varies with the dose given. Now, how to stabilize glycemia in type 1 DM? This is how Professor Unger proposed to stabilize the glycemia of type 1 DM. Say you have this glucose volatility pattern, then you want to reduce the hypoglycemic episodes by reducing exogenous insulin to 0.2 units twice a day, and this is in the level of mice dose. However, when you reduce insulin, okay, you may reduce hypoglycemia, but you will further increase glucagon level. And you don't eliminate the surge for the surge of hyperglycemia. One remedy is to use a glucagon suppressors other than insulin, and perhaps then you can change and duplicate the normal glycemic pattern by using glucagon suppressor. And again, this is again by hormonal treatment. This is in a non-obese type 1 mice uh, given two hormones versus just one insulin monotherapy. Okay? uh, Comparison of blood glucose level in a non-obese type 1 DM mice treated with 0. Unit per day of insulin or 0.02 unit per day of insulin plus leptin. Remember that leptin has been shown to decrease glucagon level. Injected twice a day. Okay, so you can see the optimal insulin curve in red and suboptimal insulin dose plus leptin, a glucagon suppressor infusion in orange. Using leptin as a glucagon suppressor leads to a disappearance of the spikes and the hypoglycemia dips. So let's look now the data on type 2 DM. Okay. Now, um, as you can see, the plasma glucagon, insulin, and glucose level in response to a large carbohydrates meal in a subject with normal and type 2 DM. You see the differences. I don't need to discuss that. And among the etiologies postulated for the pathogenesis uh, are many. Uh, It could be genetic defect, insulin resistant, beta cell exhaustion, glucotoxicity, obesity, and even redox reaction. Although any or all of these processes are plausible and may play a causative role. There is another possibility that seldom mentioned, the lipotoxicity of the islets. Now, but recently it's more and more being uh, mentioned, uh, the lipotoxicity in the islets driven by excess dietary carbohydrate consumption. The slides look at the pancreatic islet fat content in diabetic-prone DB mice and lean wild-type mice. For the first 8 weeks or so in their lives, okay, diabetes-prone DB mice in blue maintain about the same amount of body fat as lean wild-type mice. The lower panel, just before the 8 weeks mark, the pancreatic islet fat content, the white square, of the DB mice begin to rise compared to that of the wild-type mice. As the islet fat increases, diabetes uh, developed in DB mice. As the increase in fat accumulates in the islets, the morphology of beta cell changes markedly. During the first 8 weeks, okay, beta cells in the DB mice have normal appearing mitochondria and plenty of insulin granules. About 10 weeks, many of the beta cell mitochondria appear severely damaged and the insulin in the beta cell has degranulated. But does this happen in human as well? Because these are all animal experiments. Now, the graphic shows it does. No? Uh, okay, Linkway et al. Used magnetic resonance spectroscopy to determine the pancreatic fat content in human subjects and compare it to their degree of glucose intolerance. You look at the normal weight, okay? Glucose tolerant subjects were compared with obese, glucose tolerance and obese, and glucose intolerant subjects. Okay, as is apparent, there was a substantive increase in the pancreatic fat when moving from the obese glucose intolerant situation to obese glucose intolerance. Even subjects fully developed type 2 DHEN, pancreatic fat, and those with just glucose intolerance have little more, if any, more pancreatic fat. Okay, so could type 2 DN be due to insulin resistant and alpha cells? It shows here normal pancreas alpha cells are relatively sensitive to insulin. Diabetes much less sensitive, as you can compare the two. Normal with 3 picogram compared to type 2 DM with a very low 0.02 picogram. Okay, so incubated normal rat islets with the level of palmitate converted to ceramide. Normal in blue and red in DBDB mice. Okay, and this predisposes to diabetes. Greater incorporation and accumulation of ceramide is the key or the culprit for the insulin resistance. Now, we go now to the paracrinopathy of type 2 DM. The graphic on the left shows the normal paracrine response to an increase in glucose perfusion in the normal non-DM animals. As the glucose perfusion in the animal triples from 5 millimole to 15 millimolar, it is accompanied by a sharp spike in insulin release and quickly tapers off. The spike lasts but a couple of minutes and represent only about 10% of total insulin release. This rapid spike hitting the alpha cells markedly reduces the secretion of glucagon. The important things to note is that it is the spike in insulin that immediately lowers glucagon, while the lower insulin level following the spike is able to maintain lower glucagon level. Now consider what happened when the glucose perfusion is tripled in the pancreatic in the pancreas of the diabetic rats. In the graphic on the right, the most notably feature is the absence of insulin spike, the first place insulin secretion, and lack of suppression of glucagon. And in fact, insulin level increase in diabetic animals to higher level than they do in a non-DM animals. But glucagon is not suppressed and continues to rise, as you can see. Now, this is what you see in type 2 DM or the same experiment. The first thing you note is the last of the first phase insulin release the spike, but the insulin level are higher than normal overall, and because of insulin spike is gone, there's no more suppression of glucagon despite the high level of insulin. Glucagon level is also high because they are insulin resistant, and that results of a low insulin glucagon ratio of 0.4 or less than 1 tells the liver don't store in the incoming glucose and continue to manufacture or synthesize glucose to keep synthesizing glucose because we are starving, even though a big meal have been just ingested. In other words, a wiring information is being transmitted to the liver, and this is why glucose tolerance is so abnormal in hyperglycemia persists. And this is the final experiment in the series, normal mice, far left, okay, with DBDB with positive receptor, DBDB with negative glucagon receptor. Okay, these are three animals, no? Normal, DBDB mice with the presence of glucagon receptor, DBDB mice with negative or knockout glucagon receptor. What are measured here are plasma insulin on your left, and on your right is glucose. For normal mice, you can see low insulin level and normal glucose level. In the middle, DBDB mice with the glucagon receptor showed increased insulin and glucose level. Okay, uh, and um, the next is DBDB mice with knockout glucagon receptor, meaning uh, mice that is diabetic but without glucagon receptor showed similar pattern with normal mice. In the middle, the DBDB mice with the positive glucagon receptor, uh, it represents a perfect picture of insulin resistant, okay, with both plasma insulin and elevation of blood glucose. And when they injected adenovirus containing glucagon receptor to bring back the glucagon receptor, the mice developed similar pattern with insulin resistant mice with blood glucose approaching to 600 insulin level sky high. So this animal experiment showed that glucagon receptor deficiency prevents insulin resistance and type 2DM in a DB mice and injecting it back with an adenovirus glucagon receptor restore the insulin resistance. So study tell us that glucagon action is required in type 2DM. It it also tells us that insulin resistant type 2DM giving more insulin may not be beneficial but may even be harmful. So to summarize the role of alpha cells in the pathogenesis of type 2 diabetes, let me try to re-explain the normal paracrine effect. A, we see the microscopic islet cells, okay, of a human islet. Human islet shows an extensive juxtaposition of insulin and glucagon containing cells. Note that clusters of insulin containing cells in red are surrounded by glucagon containing cells in green, which seems to line in the blood vessels. B. Demonstrate the reciprocal responses of insulin and glucagon to 20 milligrams per deciliter changes in the concentration of glucose perfusing the isolated pancreas of rat. Note the first phase insulin secretion as a spike, followed by a second more prolonged period of insulin secretion. The dotted line comprises the first phase insulin secretion high enough to inhibit glucagon release from the juxtaposed islets. C. The range of plasma glucose level in a normal individual. Even extreme calorie stress or caloric stress do not raise or lower glycemia above this narrow range. Okay. Intensive exercise is marked by a gray arrow on top. Oversized meal ingestion is marked by black arrow in the lower panel. Now, let's look at type 1DM. This is the morphologic, parakinenological, and clinical features of pancreatic islets in type 1DM. A. The confocal microscopy of islet from a patient with type 1 shows replacement of insulin-containing cells with glucagon-containing cells in green, devoid of contact with beta cells. B. The absence of juxtaposed insulin-secreting cells, basal glucagon levels are increased, and they increase further in a paradoxical response to hyperglycemia from a carbohydrate-containing meal, thereby worsening the basal hyperglycemia. C. Even in the subject with optimal control type 1DM exhibited severe glycemic volatility. So, morphological, paracrinological, and clinical features of pancreatic islet in type 2DM. A. Islet morphology in type 1DM does not appear to differ grossly from the normal islets. The juxtaposition of alpha and beta cells appear to be approximately normal. Okay? B. Despite the juxtaposition, the alpha cells are not suppressed by the hyperglycemia. This appears to be secondary to loss of the first phase pancreatic spike of insulin release immediately after the ingestion of carbohydrate-containing meal. Without this spike, believed to be the cause of glucose-induced glucagon suppression, glucagon remains unsuppressed and elevated relative to the ambient blood glucose. The failure of glucagon level to decline during carbohydrate feeding exaggerates the postprandial hyperglycemia. Okay, the last one, despite the relative hyperglucagonemia and hyperglycemia, glycemic instability in type 2DM is not a problem. So, with that, I would like to close my presentation on the role of the alpha cells in the pathogenesis of diabetes by showing these slides as a guide to the alpha cell course. To understand alpha cells better, it is to know and understand all about synthesis, secretions, and actions. And synthesis is there's a gene in the cells that uh, uh, that is present, which is called the pre-proglucagon genes that synthesize a large amino acid structure called proglucagon genes, and there is a pro-hormone convertase on the specific tissues that convert or cut this long chain amino acid to a certain hormones, like for example in an alpha cells, it split a 29 amino acid called glucagon. Secretion, once its glucagon is released, okay, once the alpha cells, there are a lot of stimulants, okay, or inhibitors of alpha cells. There are a lot of processes that stimulate glucagon release. It could be paracrine, autocrine, neural, endocrine, etc. And the last one is action, which is quite important in therapeutic potentials, because the action is based on the receptors that are present in the target cells, in the islets, liver, heart, adipose tissue, etc. The most important is the liver in terms of glucose homeostasis or hyperglycemia as the cause of as the effect of glucagon, and the signaling pathway and receptor called glucagon G protein coupled receptors and the signaling pathways. So these are important things to understand, especially when we go to the potential therapeutics. Okay. So with that, I'd like to thank everybody for listening.